All right, while that's preparing to join and people are jumping in, hey everybody, thanks for joining with us. We have a super special surprise for you today. So our networking month this month, we're joined by Deb Hugers and Dr. Brooke Leverone from Epigenetics. So I'm going to do a quick background on them. So Deb is a co-founder of Epigenetics RX and La Vida Comning, both Deb and Brooke are in San Diego. So she's also a member of Exocell Bio, the former chair of the board of trustees for Bastyr University. Dr. Brooke Leverone is a naturopathic physician, a clinical educator for epigenetics, and adjunct professor for Bastyr University. And she helps patients with menopause, andropause, hormone balances, bioidentical hormone replacement, energy, sleep, and moods. And today we're going to talk about, I'm going to turn this over to them, obviously, but getting to the root cause of your client's health and wellness issues, but understanding their unique genetic blueprint and how you can really differentiate your practice by incorporating these precise personalized prevention using data and DNA. So Deb, Brooke, I'll let you guys take it from here.
All right, thank you, Riley. We're so excited to be with you today. I've been a healthcare executive for over 40 years, and I have never been more excited about this emerging science called epigenetics. So we call it the science of change because genes turn on and off based on environment and lifestyle. So what we're going to do today is run through a couple of objectives. If you want to go to the next slide, Brooke. And what we're going to be doing is reviewing with you several things that you will learn about the science of epigenetics. So the first thing we're going to do is kind of go at a very high overview. I'm going to kind of give you the 10,000 foot view, and Dr. Leverone is going to dig in. We're going to show you how to integrate genetics into your practice and how you can become an Epigenetics RX certified coach. What we've been doing is we've spent the last 6 years building the science and the protocols around this, and we're thrilled to be offering it to everybody at Sofia Health. So next slide, Brooke. So what is epigenetics? So again, you are going to see a short video that's going to talk to you about the difference between genetics and epigenetics. And so Brooke, if you want to go ahead and push play, we'll talk about it on the other side.
Ever wonder how you became you? Imagine you had a secret identical twin, which means your DNA or genetic blueprint is identical to you. Both of you.
Wow. Oh. It crashed. My system crashed.
Okay, here, I'll pick it up from here. You want me to go over there?
Yes.
Okay, here we go. We'll try it again, team. Thanks for hanging in there with those technical difficulties.
Hey, Deb, are you sharing your screen?
Yes, can you not see it?
Ever wonder how
No.
You can't see it? Sorry, guys. Let me share screen. Hang on. There we go. Let's try it again. Okay. Can you see it now?
Yes.
Ever wonder how you became you? Imagine you had a secret identical twin, which means your DNA or genetic blueprint is identical to you. Both of you inherited your DNA or genome from your mom and dad, and guess what? Your grandparents too, which makes you uniquely you. However, your DNA is only half of the story. Turns out there's another layer called the epigenome. The epigenome is above or beyond the genome, and it evolves and changes based on you and your choices in life. Epigenetics is the science behind how genes express or turn on and off based on their reaction to the environment. Here's how epigenetics works. Imagine your secret twin is raised by another family and makes different choices than you do growing up. Your twin has a stressful job, smokes, loves sugar, and watches TV all the time. You, on the other hand, love to run, meditate, and eat from your garden. These lifestyle choices modify your genes and create your personal epigenome by creating epigenetic tags that sit on top of your DNA. These epigenetic tags tell your genes what to do through methyl groups. They tell which genes to do what and how much. Methyl groups can instruct genes to turn on and off. Good lifestyle choices turn healthy genes on and unhealthy genes off. Bad lifestyle choices turn unhealthy genes on and healthy genes off. Your DNA is wrapped around proteins called histones. When your DNA is tightly wrapped around the histones, the genes are unreadable and not expressed. When the DNA is loosened around the histone, your genes are accessible and can be expressed. Look at it this way: your story is written in pen and pencil. Things in pen you can't change. That's in your DNA. Things written in pencil you can change. That is epigenetics. Mutations or SNPs can be viewed as typos in the copying of cells. Mutations aren't bad, they just make you unique. Genetic mutations can affect how your genes function, just like throwing a wrench in a moving set of gears. Where is the wrench impacting your health? In many cases, adding the proper doses of methyl donors with your nutritional and lifestyle choices, along with supplements and/or prescription intervention, can have a positive impact on your health. Do it for you and your future generations. Talk to your practitioner about a precise, personalized treatment plan made just for you. For more information, join our community at epigeneticsrx.com.
So thank you. Sorry for the technical difficulties, but what we wanted to do is we wanted to share how the epigenome is different than the genome. So what's really important about what we want you to learn today is that you basically inherit your DNA, and that is your genetic blueprint, but you also, on top of your DNA, have an epigenome, and you can also inherit that for up to 18 lifetimes. But the good news is you can change it in one, because the environment basically affects your gene expression. So your grandparents and your parents count, and even your great-grandparents count. But when you begin to look at diet and lifestyle, you can see that you can really change your gene expression. So what we want to do is we want to really look at your health span, and we want to increase your health span by you looking at your genetics and really understanding what's happening inside of you. So one of the things that we're going to talk a little bit about today is how we can reverse those bad epigenetic tags that are attached to your DNA through methylation, and how the science relates to that. So obviously you're treating a lot of patients, you're supporting them either in their weight loss or their psychological issues, their motivation issues, and there may actually be biochemical and genetic things that you're not aware of that we can help you with and help you get your patients and customers to the right level. So one of the things we like to talk about is the epigenetics of royalty. So all of us want to be queen bees, as we always talk about. So we talk about the queen bee's royal jelly. So what most people don't know is all bees are born with the exact same genetics. But what makes a queen is the royal jelly that she's fed when she's a young bee. So what it really relates to is the epigenetics and what her environment of that queen bee had when she was basically treated differently than all of the other bees and was fed queen bee royal jelly. So what we know, not only from these animal studies, which Dr. Leveron will talk to you about with rats, is that the environment really does matter. And that's what the science of epigenetics is about. And that's why we believe this is such an important science for everyone at Sofia Health to be tied to. So Epigenetics is the science and the evidence behind lifestyle medicine. So it's really simple. Changing your diet and lifestyle changes your epigenome or the epigenetic tags, which changes your gene expression, which literally changes you. So what we're talking about today is understanding some of the things you might not be able to see through the science of epigenetics. So what is really exciting is that we're going to teach you how you can turn the good genes on and the bad genes off, and make sure that your genes are expressing based on your lifestyle and environment. So with that, I'm going to give it to Dr. Leverone to really dig through the science and talk to you a little bit about what does methylation mean and how does that affect the environment and how genes turn on and off. So Dr. Leverone, would you like me to run the slides, or you want me to switch it over to you?
No, let's run the slides. I realized I think there's something up with my system that doesn't allow me to do full it crashed when I had to do a full presentation last time. So
great. So take it away.
For technical, I'll let you run the slides. Hi, everybody. Thank you for joining us today. I love that you're here. Deb and I love talking about all of this information. And for me, what doing genetics has done has really been allowed me to literally change the way that I practice medicine, and has allowed me to help people in such a different way, especially with my different cases, my difficult cases. And so we're going to talk a little bit about how you can implement that. And one of the things is that looking at the DNA as a blueprint and looking at what people may be susceptible to, and therefore maybe we need to focus a little bit more of our attention in those areas. But as Deb was saying, and some of the information that we saw earlier in the presentation, is that we have the ability to change the expression of our DNA. And that, again, has to do with the epigenome that's wrapped around the DNA that opens and closes the DNA for gene activation. And this is controlled in large part by what are called those methyl groups. And partly looking at the DNA also means, does the patient actually make methyl groups? Because some people have a difficulty making methyl groups. And some of you may have seen this before. This is that MTHFR gene that a lot of people will look at and see if people are making or not making. So some people have a hard time maybe making these methyl groups that are so important to genetic expression. Some people may actually have an easy time making methyl groups, but the body's ability to process these methyl groups in other areas may be compromised, and then they're a little bit sensitive to too many methyl groups. So what do you do? How do you know if you need to give somebody methyl groups or not give somebody methyl groups? Because as we know, they're very important, but in some cases, as you can see on the slide, some patients can be actually sensitive to the methyl groups, and we can have a whole other set of problems as a result. And being able to know what nutrients are appropriate for maximizing their health and wellness can be a really powerful tool in really personalizing their treatment plan and helping them along their wellness journey. So Deb, you can go to the next slide. So again, from the slide, we always want to treat the root cause. So, when we're with the root cause, we don't want to just give a prescription or even just give supplements to be able to take somebody's symptomology away. We want to actually know what is going on. Why is the patient having the symptoms that they're having? Why are they suffering? What is going wrong in their unique biochemistry and physiology, all the way down to their DNA and the gene expression, which genes are activated and not activated, and what enzymes are working and not working. So that's the foundation there, the assessment there, and having the genetics is a huge part of that. So we can jump to the next slide. So when we see that somebody has that MTHFR gene that I was just talking about, we can see that some patients have a genetic variant. So backing up a step, when we look at DNA is the double helical, right? Double helical DNA strand. So we got one copy from one parent, one copy from the other parent. Both copies are a mirror image of each other. They're the same genes on each side, but one's from one parent, one's from the other parent. And your body has a 50% chance of reading one blueprint versus the other in order to make the proteins or the enzymes that day. So if one of those copies is not the right copy and there's a genetic variant, or a gene SNP, as we call it, then we say that you have a heterozygous mutation, which means that you are down about 40% loss of function in that gene's ability to do its job. If you have both copies of the DNA with a mutation, then you're down about 70% of the function of that gene. There are two different gene locations or SNP locations for the MTHFR gene. That's the C677 and the A1298C that is associated with function of that enzyme. And if you have a heterozygous mutation of both, then you're down about 50%. So knowing what the level of function is can be really important, because it's like, oh, do you need a little bit of methyl groups? Do you need a lot of bit of methyl groups? And how is that sort of interplaying? We can jump to the next slide, Deb. So when we look at the genes, I always think of the system as one big biological factory with all these assembly lines on it. And each of these assembly lines has a machine that converts one product into the next product. So I always think of the MTHFR gene as that first assembly line where we're converting folate into methylfolate. And then that methylfolate is really important because it goes to all the other assembly lines in the factory, kind of like gears on a machine, which is what this schematic is telling us. So if you go to the next slide, Deb, where we can see where there's a lot going on here. So this can be a little bit overwhelming. And I admit, I've been studying genetics for over 10 years. And in the beginning, it seemed like it made sense, but look, there's so many moving parts here, right? So luckily, Epigenetics has created a really great model to help make it easy for you. We're simply trying to break down, does this person need methyl groups, or are they going to be methyl sensitive because some other areas in the factory aren't working? Maybe some of the other assembly lines can't handle those methyl groups. So maybe they're not working and maybe they're going to fall into that sensitive category. So what our job is to try and help break that down and make that really easy for you to understand, so that you can then tell your patients, oh yeah, you do need more methyl groups. You do need less methyl groups. So that's what we're hoping to do. So if we go to the next slide, you can see what our report looks like. So on your report, this is the result, and you can see there's a lot of different data points here, but you can see some are homozygous, some are heterozygous, and some are wild type, or meaning, you know, the natural variant that doesn't have loss of function. So when you get these reports, then we can, if you go to the next slide, we can see where those genes are showing up. So we can see the different machines on the different assembly lines circled here that aren't working for that patient and that are throwing wrenches in the system. So the wrenches in the system, again, stop all of the machines in the factory from being able to move forward. And that can show up in lots of different ways, depending on their certain gene layout, what kind of genes are coming into play. So, but using the schematic and using our training, again, we break it down so it makes a lot more sense. It's as simple as literally thinking about that factory with all those assembly lines and thinking about which machines are working and which machines aren't working, knowing what product that machine is making versus that machine. And then you can kind of figure out, oh, okay, I need to tune that machine up and turn that machine down. So it does get a lot easier when it's sort of laid out in an easy to digest paradigm. And it gets less overwhelming over time, I promise. All right, we can go to the next slide here. So the reason we focus so much on the methylation component is because we know that there's so much that the methyl groups do. So again, the methyl groups have function in turning on and turning off genes, which if there's certain genes that are meant to be off and they get turned on, that can be a problem, and vice versa. So we also see in that all those assembly lines in the factory and that gear work schematic we just looked at, those methyl groups can drive a lot of other functions too. They can drive detoxification and enzyme production and tissue repair and DNA protection. Really, really important for fetal development. This is happening all the time. So it's a really important component to health and wellness, and understanding it can make a huge difference. So we've got some studies here to sort of implement or to sort of, drive that point home, if you will, where we're seeing this show up. So jumping to the next slide, this is a schematic of when we see lots of methylation. So there is the DNA structure in that top left corner of this diagram where you see all the little green bubbles. Well, these are basically, these are all the methylated histones. So we've got all the methyl tags that are controlling the DNA. As a result, there is a specific gene that gets turned off, and we can see there's very little of these green fluorescent proteins showing up on the microscopy and looking under the microscope at these gene samples. So we have low activity of gene expression. In other words, decreased gene expression of this specific gene because we have healthy methyl groups holding the DNA together. Then on the next screen, we see that there's not a lot of methylation happening in the histones, so not a lot of activity there. And as a result, this gene is turned on, this inappropriate gene is turned on. We can see all the phosphorescent protein tags being showing up in the microscope, where we can see that there's increased DNA activity. And so it's really simple when they do this. If you go to the next slide, we can see that there's, through lab testing in these cells, they basically added methyl groups to see the genes get turned off, and then they gave another sample, a medication that took the methyl groups away, and we see that gene get turned on. So it's very clear. We can see that in basic biochemistry that we can see, and microbiology, that we can see genes get turned on and turned off directly as a result of enough methyl groups or no methyl groups. So it's very clear in the science. It's very powerful stuff that we can see in some of these basic models. This can come into play and be really important in the next slide, where we can see that certain genes need to be turned off and certain genes need to be turned on. So if we have uncontrolled growth, if we have certain uncontrolled growth that can happen because certain genes get turned on, and we have normal growth when certain genes get turned off. So obviously this is important when we're talking about cancer growth and tumor suppressor genes. So tumor suppressor genes can be can be regulated by the methylation process. And when we have inappropriate methylation of genes that are being turned on and turned off, we can get inappropriate cell growth as a result of that as well too. So in the next slide, we're going in to talk about the some of the studies that we've seen in the rat model, where we've seen both the
There we go. Come on.
There we go. So
we
we've seen, there's been lots of studies in this rat model about healthy methylation and unhealthy methylation and your sort of environment. So again, one of the things that we want to work on with patients is controlling their genetic, not controlling their genetic sources, but optimizing their wellness, which we can do by optimizing their genetic expression. And we're seeing that that can start as early as the womb and in early childhood, but also we can inherit these gene markers that can go back 18 generations. So what we mean again is that the methyl groups on the epigenome, again, control what genes are actually being turned on and turned off. And some genes need to be turned on and some genes need to be turned off, and appropriate methylation controls that. But the other thing that we see is that lifestyle and behavior also control that, which is very fascinating. So healthy diets will actually affect the genetic expression. Stress management will affect the healthy genetic expression. Lifestyle, exercise, minimization of toxins. We see toxins and oxidative stress negatively impact gene expression. So there's been lots of these really interesting studies in some of the rat models where we have a mouse model, where the mama mouse is licking and nursing and coddling and caring for its young pup. And as a result, the young pup grows up and is very calm and relaxed and healthy. But we have other moms who don't nurture and don't care for their young pups, and they're sort of ignored, and they have increased risk of anxiety. And part of this has to do with the activation of the glucocorticoid receptor. So we can move on. So the GR raceme, the glucocorticoid receptor gene, is a feedback loop for stress management that helps individuals and all of us to be calm and manage the stress response. So again, so now we're seeing that the behavior with the mom is positively or negatively affecting the outcome of the baby pup. And from a genetic level, we're seeing why, because we're seeing inappropriate genes, certain genes being turned off that shouldn't be turned off. And when we go to the next slide, we see that mama is caring for the baby and there's more activity, the low activity, the high activity, there's good activity with the mama, and that gene is now being turned on. And so we're having the glucocorticoid receptor gene turned on. And that receptor gene is a feedback gene for stress management. So it allows the body to reduce the cortisol expression in the stress response. So on the next slide, there's just a brief schematic that talks about when the kidneys and the adrenals release the cortisol that feeds back to the brain, and these glucocorticoid receptors in the brain give a feedback that says, hey, that's enough cortisol, let's everybody calm down now. But if you don't have those receptors to mitigate that response, then you're going to be prone to anxiety. So again, this comes to lifestyle activating or suppressing or activating an important gene. So again, we want that gene turned on, but in a stressful situation, that gene is turned off. So these rats are more prone to a poor stress response as a result. In the next slide, we're seeing, so in that particular example, the methyl groups, there was decreased methylation of the gene, which allowed for it to be turned on. We wanted that glucocorticoid receptor to be turned on to manage the stress response. And healthy nurturing actually reduced the methyl around that particular gene. What's fascinating is, this is sort of what Deb was talking about in the beginning, is we can change genetic expression. So if we were to, in this particular example with these rats who grew up in a highly nurtured environment with mom, and they had that glucocorticoid receptor gene turned on, the body didn't methylate that gene appropriately so that it could be turned on. But we suddenly take that same rat and then we inject a bunch of methyl groups, then that same rat now can become really anxious because now the glucocorticoid receptor gene is turned off and they don't have the feedback to calm themselves down. In clinical practice, we can see this in some patients who would be called methyl sensitive, where they don't respond well to high amounts of methyl groups. And this is helpful when we're looking at the genes, like, oh, do you respond well to methyl groups or do you not respond well to methyl groups based on your DNA and based on your environment? So we do know that we can reverse this genetic expression in some of these rat models that we've seen, which is highly fascinating stuff.
So Dr. Lebaron, is this just a really good example of how you can give somebody the wrong B vitamin? So just saying take B12 and you can make them more anxious by giving them the wrong kind of B12?
Yeah, exactly. And this is what I've seen a lot in my clinical practice, is that the MTHFR enzyme has gotten a lot more attention in the last 5-10 years. And I've seen a lot of patients come to me who saw another practitioner and they had maybe a single heterozygous mutation of one of their MTHFRs. And that practitioner was like, oh, you don't methylate well, you need a lot of methyl groups. They give them a bunch of methyl groups and now the patient is anxious and can't sleep and having all these other issues. Well, they didn't need that many methyl groups because maybe they had other issues in the pathway and they were only deficient by about 40% at the most. And so being able to understand which vitamins the patient needs and can tolerate is a big part of the picture, which is why you need to know more than just the MTHFR. You need to know everything involved, because it really affects so many things, from DNA expression to just day-to-day function. So this is so a pup that's raised by an anxious, low nurturing mother can become an anxious adult, or a pup that's raised by a relaxed, high nurturing mother can become a relaxed adult. So that's sort of, what we were talking about initially. And this is, The study is commonly referred to as lick the rat study, because that's the bonding that the mother does with the pup. You know, she's lots of groom. You know, we've all seen animal videos, and a lot of us have had animals and kittens. The mom always grooms them. So the bonding again, so this is just sort of reiterating some of the things that we talked about, affects the methylation pattern of the gonadotropin receptor gene. And that has a long-term effect on the personality type, which is fascinating. So you think about, you know, and this is things that are seen, you know, they're fairly intuitive in our culture when we see on the human level, right? Somebody that grows up in a high trauma, or, you know, doesn't grow up with a parent, or, you know, foster home, you know, they're going to be more prone to anxiety. And part of that is, you know, their cultural upbringing behavior models. But we're seeing, at least in the rat model, that part of that is the effect that the environment had on the methylation of that particular gene. And the healthy methylation patterns are affecting these expression of these genes that affects their personality in these rat models throughout their entire life. So it's not just, you know, in those early years when they're under the mother's care and getting all the love, but their whole life, we're seeing that they continue to maintain the relaxed mentality, just because those early years they got that nurturing that affected their DNA expression, which is, you know, pretty powerful stuff. So if any of you are working with women who are trying to get pregnant, and, or, you know, maybe some pediatrics, understanding this information can really set the mother and the new baby or their young child up for great success later. You know, you're really optimizing their health and wellness when we can utilize these modalities. So we have, so speaking of our pregnant mothers, so another component of this particular study was what's called the agouti mouse. So it's looking at the agouti gene, and they're looking at this pregnant mouse with the agouti gene. It was given lots of methyl groups while it was pregnant. And the agouti gene basically is a gene that causes increased risk of obesity and diabetes. And in these mice that had the agouti gene, when they were pregnant, the mother was given all these methyl groups, and that ended up turning off the gene in the babies, so that the babies all had normal skin color, no diabetes, no obesity. So we can see in this next slide, these two rats are genetically identical. They're genetically identical rats. But you can see the one on the left, that agouti gene was turned on, and the one on the right, the agouti gene was turned off. I mean, and that's a huge difference. They don't even look like the same species. But we see that this poor guy has diabetes, obesity, and change in its fur color as well, too, as a result of the activation of that gene because of decreased methylation. So again, in the next slide, we're seeing that the methyl groups are appropriately turning off this gene so that we have a healthy mouse. But in the mouse that didn't have the healthy gene, or excuse me, didn't have the methyl groups, they expressed that gene. And so they're more prone to obesity, diabetes, and cancer too. I forgot to mention that they are higher prone to cancer as well too. So again, in the next slide, so then the other component is that the pregnant mom was then fed BPA, which I know, I'm sorry, animal studies are terrible, but the pregnant mom was fed BPA, but then different diets. So both the moms of these mice in this scenario were exposed to BPA, but one mom was given the choline, the folate, the B12, the excuse me, betaine. These are all methyl donors and healthy nutrients for DNA expression. So even with the exposure of a toxin that can activate this gene inappropriately, providing the right nutrients can still turn off the gene. So the other mouse did not get the supportive methyl groups, and so that gene did not get turned off, and they were at higher risk for obesity. So it's totally reversible. In other words, genetic expression is totally reversible with the right nutrients.
Yeah,
so again,
doctor, I think this slide to me, I get the animal studies, but if these two rats are genetically the same, and it just shows how much diet, lifestyle, and nutritional intervention. I mean, everything that the Sofia Health group does is really represented in this slide.
Yeah.
Yeah.
Yeah, exactly. And what's so cool to me is that it's, I feel like some of the statements of, oh yeah, healthy diet, healthy lifestyle, seem sort of basic in concept and intuitive, especially for all of us who are healthcare practitioners. But when you can see how deeply important that is on the genetic level, it's very powerful. And knowing that we can take that up to the next level with the right nutrients it's so cool. I mean, I love when my patients, I give them the right B vitamin and they come back a few weeks later, they're like, oh my God, I feel so much better. Like everything's better. And I'm like, really? I just gave you B vitamin. I mean, and even though I can look at the research and be like, well, that's how it's supposed to work, and I understand the biochemistry behind it, it still fascinates me when it's that simple. And it's just such powerful stuff. I just love it so much.
It's rewarding.
So the challenge with, so it's a little bit easier in the rat population to study these things because, we've got the rats in the lab and, they poor things, they've lived there for generations upon generations. So, we can study all of that and we can control what toxins they're being exposed to and all those kinds of things. But of course, there's obviously going to be some ethical, like we're not going to study a pregnant human woman and inject her with BPA and see what happens. So there's some challenges. Obviously, we can't, fully study this all the way on the human level. Additionally, there's other complicating factors because we have the, multi-generation. So if your mom is the first generation, the female fetus is the second generation, or the male fetus is the second generation. And then their eggs or sperm is the third generation. So there's multi-generations that are being passed along here. And that, again, that's the DNA, any random mutations in the DNA, but also the epigenome, which is where the methyl tags come in, that's wrapped around that DNA that controls the expression of those genes. And we can trace that back, at least like just by sampling the DNA of family lines, up to 18 generations, where we're seeing that epigenome pass on from generation to generation to generation. So it's a little bit harder to track in the human model because of these variabilities, but we're seeing obviously lots of good research that is explaining to us what's going on. So we know that there are certain conditions that are directly affected by methylation. So, you're thinking about, I was talking earlier about those tumor suppressor genes. Methylation is supposed to turn those off. Non-appropriate methylation means those tumor suppressor genes are supposed to be on, Inappropriate methylation can turn those off. And now you don't have tumor suppressing activity happening. And so you get cancer development. Cardiovascular disease, huge in the methylation. Autoimmune disease because of immune expression on the DNA. So when we have not only the methylation because of DNA expression, because of tumor suppressor genes coming on and off, but we also know methylation affects things like homocysteine levels and vascular health with hypertension. We see methylation affecting all those things. We can have methyl groups that are needed for day-to-day detoxification, so that can affect certain chronic conditions. We see methylation huge in Alzheimer's. So there's a huge genetic component with Alzheimer's and methylation. And autoimmune because of the way that the DNA controls immune function, and methylations play on that as well too. And then of course, diabetes, which has to do with liver function and blood sugar control and multiple other factors. But you can inherit these genes, and the methylation turning them on and off can impact your risk of diabetes as well too. So when we do at Epigenetics, when we are doing our genetic panel, again, the MTHFR is not the end all be all. It's literally the tip of the iceberg. So when we were looking at that schematic earlier with all the gears turning, everything started with the red gear in the center, which is that methylation primal pump, which causes the methylation of folate to methylfolate. And then that methylfolate goes and supports hundreds of other functions in the body, including that DNA tags and other areas. So it's important to see how all those interplays are connected in order to know, is your patient going to respond well to extra methyl groups to support their health and well-being, or are they going to be sensitive to those methyl groups and be more prone to anxiety? So we look at the full complex methylation panel, which is the MTHFR gene, which again makes the methyl group to begin with. And then the MTR and MTRR work together to donate methyl groups to homocysteine to make methionine, which makes SAMe, which does the methylation tags on the DNA. BHMT also helps to make homocysteine into methionine to make SAMe. These also play a role in reducing cardiovascular risk. And then we look at CBS genes that are a component of one of our detoxification pathways. And if somebody has a problem in this pathway, they're not going to be able to handle a lot of the CBS, or excuse me, they're not going to be able to handle a lot of the MTHFR, the methylfolate coming in. And so that's where we have to go, oh, this person also has a CBS, so let's be cautious with giving them too many methyl groups. And then we also look at vitamin D receptor genes, because this has a huge effect on moods, energy, your vitamin D levels, which affects bone and immune health, skin cancer risk, etc. So looking at the vitamin D, CUMT and monoamines. These are other enzymes that are really, really important for daily detoxification and neurotransmitter metabolism or balance, which can affect moods and energy and psychiatric conditions. Issues in these genes, if you have these gene mutations, the patient cannot handle a lot of methyl groups. So you got to be careful. This is where we're going to get that increased anxiety from the patient if we give them too many methyl groups. So when you can see all of the genes together, then it gives you a much better perspective on what the patient actually needs, how that's impacting their day-to-day health and wellness, and then making sure that you're optimizing their function in the right way that's not going to overstimulate them. And these, I could go on and on about how much this has helped my patients in my practice, but I'm hoping that you guys can all see where this can play a role in really digging into that deeper level of that diet and lifestyle nutrition, where we're really optimizing genetics expression, but also we are supporting their day-to-day metabolic functions in a way that works for them so that they can feel their best, and that we're doing it appropriately without just willy-nilly throwing methyl vitamins at them or other things at them that could make them worse potentially based on the rest of their genetics, not just the MTHFR.
Yeah, doctor, I think the one thing that I always take away when you talk about this is just looking at MTHFR is just not enough. In fact, when we started this 6 years ago, that's what we thought. But then we realized the interplay between COMP and CBS and all of the others really created a whole nother story. So it's really important to get a good picture, a foundational picture.
Yeah. It makes such a difference. So, everybody's going to be a little bit different, both in their DNA and their epigenome, because what was their diet and lifestyle and their growing up. I've had patients come to me and their genetic profile, they had so many genetic variants on there that I thought, oh, for sure, this patient's going to have anxiety and digestive issues and allergies and skin and joint pain. And they're like, no, I'm chill. I feel great, And so it's very powerful when we can see how much a healthy environment really does have a direct impact on these gene expressions. So even though the DNA blueprint looked like they would have been a hot mess, their diet and lifestyle allowed for them to function optimally and feel great, despite what their DNA says, because the body was able to control those genes in their favor. So understanding what the genes look like and how that can be playing a role in their symptomology on a day-to-day basis is so helpful. Understanding that you can improve the expression of these genes through diet and lifestyle, also really helpful. And it's just, like I said, this has completely changed the way that I practice medicine. It's been extremely helpful for all of them, for the patients that I work with.
Yeah. So, I mean, bottom line is, I know, because I know we're hitting up against the time frame and we want to open it up to questions, but the science of epigenetics is just so fabulous and wonderful, but it allows providers like yourself and all the Sofia practitioners to be more precise and personalized and preventive. Would those be the takeaways that you would talk about as far as personalizing the treatment plans?
Yeah, for sure. Because, if you see where their genetic risks are, then okay, well, we need to focus on this type of diet and supplementation so that we're reducing those risks. And then knowing what the whole picture looks like so that you're supporting the pathways in the best way possible and not pushing them in the wrong direction.
Yeah, that's great. Well, thank you, Dr. Leverone.